Welcome to this deep dive. Today we're getting into a really pivotal discussion from an expert forum, all focused on what's next for Alzheimer's disease immunotherapy.
That's right.
We've seen these initial encouraging results with amyloid targeting drugs after, well, years of challenges. So the big question everyone's asking is, where do we go from here?
Exactly. And we're drawing from a fantastic session titled Improving the Therapeutic Potential of Immunotherapies in 80 Can Novel Strategies, or Next Generation Drugs Halt, or Further Delay Disease Progression.
Sounds comprehensive.
Oh, it was. You had statisticians, clinical directors, top neurologists, plus key people from biotech and pharma think Pentaur Corporation, Hoffman La Roche, Acumen Pharmaceuticals, Indiana University, someone formerly at Biogen, Athena Biosciences. Really a powerhouse group.
Wow. So the mood must have been, what, cautiously optimistic? Like progress is happening, but everyone knows the hurdles?
Precisely. There's a definite sense of momentum, but also a very clear-eyed view of the challenges in actually getting these therapies to patients effectively.
Right. So our mission today is to really unpack that conversation for you. We want to pull out the crucial insights. What do these experts see as the biggest roadblocks? And maybe more excitingly, what are the future directions they're looking at?
Okay, let's dive in.
Okay, so thinking about those roadblocks,
Yeah.
what came up first? What do the experts flag as the biggest immediate challenges for, continued success?
Well, right off the bat, a major theme was this difficult balancing act. How do you effectively reduce amyloid in the brain?
Which is the goal.
Right, the primary goal for these drugs, but do it while minimizing the risk of ARA, those amyloid-related imaging abnormalities.
Yeah, ARIA, that's been the big safety concern we keep hearing about.
It really has. It requires a very delicate approach.
Yeah.
And tied into that, someone from Athena Biosciences, Chad, raised a really fundamental question. Have we even hit the maximum benefit we can get just from targeting amyloid?
That's interesting. Are we getting all we can out of this specific approach?
It's a critical question. Because if the answer is no, then maybe we need to focus on refining these anti-amyloid drugs further. But then Janice from Hoffman La Roche chimed in, reminding everyone about the sheer complexity of Alzheimer's.
It's not just amyloid, right?
Exactly. Is tackling just amyloid really going to be enough in the long run? Which naturally leads to thinking about combining treatments or maybe using them sequentially.
Makes sense. It's a complex disease, so maybe you need a complex solution, not just a single silver bullet. Eric from Acumen Pharmaceuticals brought up something really practical, too. Accessibility.
And absolutely. That was a huge point. The actual infrastructure needed, infusion centers, enough MRI scanners, the sheer workload on neuroradiologists who need to read those scans.
It sounds like a bottleneck.
It really is. Donna Wilcock from Indiana University mentioned that even major medical centers are finding it hard to scale up to meet the potential demand. And Yuval Zabar, who used to be a Biogen, really stressed the need for more than just clinical trial success. We need to see greater efficacy, better long-term outcomes in the real world.
So not just does it work in a trial, but how well does it work out there in regular clinics?
Precisely. How well can we actually implement these therapies day to day for the patients who need them? It's one thing for it to work under ideal conditions, quite another in routine practice.
So it's really two big things. Keep pushing the science forward and figure out how to actually deliver those breakthroughs.
You've got it.
Okay, so delivering them. That also involves knowing who to deliver them to, right? The discussion moved into identifying patients who benefits most, who's at higher risk.
Yes, a really crucial area. Suzanne Hendrix from Pentaur Corporation highlighted a kind of tension here. Companies are often hesitant to define very narrow patient groups.
Why is that?
Well, mainly because of concerns about limiting access. They don't want to be seen as excluding people who might potentially benefit.
Okay. But scientifically?
Scientifically, there's a real need to understand who's likely to get the best response and, just as importantly, who might be more prone to side effects like ARIA.
And are we getting any clues from the data we already have?
We are, slowly. Donna Wilcock pointed to some post-hoc analyses that's looking back at trial data after the fact from the donanemab trial. It suggested that people who started with lower levels of tau pathology actually had better outcomes.
So maybe intervening earlier makes a bigger difference.
That certainly seems to be part of the picture, yeah. And we already know about the increased ARIA risk for people with 2 copies of the APOE4 gene, the homozygotes.
Right, that's been established.
But the experts were discussing, okay, we know the risk is higher, but is the cognitive benefit necessarily the same for that group compared to others? It's still a bit unclear. And Eric Siemers from Acumen suggested that a lot of that APOE4-related ARI risk is probably linked to how much cerebral amyloid angiopathy, or CAA, someone has.
CA, that's the amyloid buildup in the blood vessel walls in the brain.
Exactly, and identifying it accurately is becoming really critical.
How do we do that now?
Well, Chad Swanson from Athena Biosciences noted that we can sometimes see signs on MRI, like tiny bleeds called microhemorrhages, but it's not perfect. We really need more sensitive, more reliable methods.
Okay.
And Donna Wilcock brought up some interesting, though still early, research. It hinted that things like high blood pressure might increase ARIA risk, but also maybe that patients already on antihypertensives seemed a bit protected.
That is interesting. Needs more research, I guess.
Definitely needs more research. But the big takeaway, the consensus among the experts, was the urgent need for better biomarkers.
Biological markers, right, in blood or spinal fluid.
Yes, not just for amyloid, but also to spot other things going on in the brain, like alpha-synuclein pathology or TDP-43, which might really affect how well someone responds to an anti-amyloid drug.
So better biomarkers seem key to making treatments safer and more personalized. What about specifically using blood tests for CAA and ARIA risk? Was there progress reported there?
There was definitely excitement about blood biomarkers for AD in general. That field's moving fast. But specifically for CAA, they stressed an urgent need for quantitative tests.
Quantitative meaning?
Meaning tests that don't just say yes or no, but can actually measure how much amyloid is building up in those blood vessels. A reliable blood marker for CAA.
Wow, that would be a game changer. Avoid some of the reliance on MRI.
Absolutely. Make risk assessment much more accessible. Donna Wilcock mentioned some early work, not published yet, looking at the ratio of A-beta-40 to A-beta-42 in the blood, and also a marker called GFP, which indicates astrocyte activity as potential flags for CAA.
Okay, promising leads then.
Yes. And Janice Smith from Hoffman-La Roche also said there's a big push to find fluid biomarkers, blood or CSF, that could predict who's likely to develop ARA before treatment starts.
That would obviously be huge for safety, being able to flag high-risk individuals with a blood test.
Exactly. It would significantly improve how safely we can use these therapies.
Okay, so let's get into the nitty-gritty of the drugs themselves. There was talk about targeting different types of amyloid. Could that affect ARIA risk?
Yeah, this is really at the cutting edge. The theory is maybe if you could design a drug to only target A-beta oligomers.
Those are the smaller, sort of free-floating clumps thought to be really toxic.
Right. If you could hit just those and avoid the amyloid plaques, especially the amyloid embedded in blood vessel walls, the CAA, maybe you could reduce ARIA.
Sounds logical, but is it doable?
Well, Chad Swanson pointed out the practical challenge. It's really hard to make an antibody that specific. Depending on the concentration, many antibodies tend to bind to both oligomers and plaques to some degree.
So achieving that perfect selectivity is tough. What about other approaches, targeting different forms?
Eric Siemers gave a good recap. Remember solanezumab? Targeted monomers, single A-beta units, very safe, no ARIA.
But didn't work very well.
Exactly. Didn't clear plaques effectively, didn't show much cognitive benefit. Then you have something like donanemab, targeting protofibrils, slightly larger clumps. It does clear plaque.
But still has ARIA risk.
Right, likely because it still interacts with CAA. But Donna Wilcock mentioned something really intriguing. Brain shuttle technology.
What's that?
It's a way to try and sneak antibodies across the blood-brain barrier more effectively, specifically targeting the tiny capillaries, maybe bypassing the larger vessels where CAA is more an issue.
So deliver the drug differently to potentially avoid the problem areas.
That's the idea. Potentially reducing ARIA risk by changing the delivery route.
That's clever.
It is. And Eric Siemers made a really important point here. The amyloid hypothesis isn't just one single idea.
Meaning
Targeting monomers is different from targeting oligomers, which is different from targeting protofibrils or plaques. The specific amyloid species you target really matters for both efficacy and safety. It's not all the same.
Got it. Okay, let's shift slightly. Combination therapies came up. What kinds of combinations did the experts think held the most promise?
There was broad agreement that just clearing amyloid, the stuff outside the cells, might not be enough. You probably need to tackle the neurodegeneration happening inside the cells too, especially related to tau protein.
Right, tangles.
Exactly.
Yeah.
So Yuval Zabar talked about combining anti-amyloid drugs with tau-targeting therapies, maybe drugs that reduce tau production.
Okay, amyloid plus tau. What else?
Donna Wilcock was quite keen on combining anti-amyloids with inflammation modulators.
Targeting brain inflammation.
Yeah, the hope being you could potentially reduce ARIA and maybe impact some of the other pathologies often seen alongside amyloid and tau. Although, Eric Siemers added a note of caution. He mentioned recent data around TREM2, an immune protein in the brain, has actually made the inflammation picture even more complex. We don't fully understand its role yet.
So targeting inflammation might be trickier than it sounds.
It might be. We need to understand it better. Janice Smith brought up practicalities, again, access issues with combinations, and maybe the need for oral drugs, small molecules, to make complex regimens easier for patients.
Sure, taking multiple infusions would be tough.
Definitely. Chad Swanson also leaned towards tau as a key partner for combination, especially given some encouraging recent results in anti-tau drug development.
Any other combination ideas?
Yes, sequential therapy was also floated. The idea is, first, use a powerful anti-amyloid to clear out the existing plaques.
Okay, clean slate.
Sort of. And then follow up with a different kind of drug, maybe a gamma secretase modulator or a base E inhibitor, to basically turn down the faucet and reduce the production of new amyloid, preventing it from building back up.
Hit it hard, then keep it down. Makes sense.
Oh.
Okay, we focused a lot on passive immunotherapies, giving people antibodies. What about active immunotherapy? Vaccination.
Right, the vaccine approach. It definitely has a strong appeal, this idea of maybe a preventative shot, a one-and-done strategy.
That would be incredible.
It would. But the experts acknowledge there are still pretty significant hurdles to overcome in developing effective Alzheimer's vaccines.
But Donna Wilcock seemed optimistic, especially for prevention, linking it to early detection with blood tests like p-tau217.
She did. The vision is identify high-risk people very early, maybe even before symptoms appear, and vaccinate them to stop the pathology from ever getting a major foothold.
But there are safety concerns, right? Lessons learned from past trials.
Absolutely. The AN1792 trial from years ago, which had cases of what we now think was probably ARIA-like inflammation, is a constant reminder. And as Eric Siemers pointed out, a key issue with vaccines is there's no immediate off switch.
Right. With an infusion, you can stop it. With a vaccine,
your immune system is activated, and you can't just easily turn that response off if someone has a bad reaction.
That's a major safety consideration.
It is, and Chad Swanson mentioned the sheer difficulty of designing trials for prevention. Following thousands of healthy people for potentially decades to see if a vaccine works, that's a huge undertaking.
Yeah, logistically challenging.
But there's optimism, too. Janice Smith talked about advances in immunology using shorter protein fragments, peptides, in vaccines to try and avoid triggering the unwanted T-cell responses that might contribute to inflammation, potentially lowering ARIA risk.
So, smarter vaccine design.
Hopefully. There's also work on understanding antibody levels, how they change over time after vaccination, and even ideas about combination vaccines targeting both amyloid and tau.
Interesting. Any downsides mentioned?
Well, one concern raised was whether aggressively targeting amyloid with a vaccine might accidentally deplete the monomeric form too much, the single units that might actually have some normal, useful functions in the brain.
Unintended consequences.
Potentially. So researchers are trying to engineer vaccines to specifically avoid hitting those potentially beneficial monomers.
Okay, so active vaccination. Lots of potential, especially for prevention, but still significant safety and design challenges being worked on.
That sums it up well.
Finally, let's talk about the real-world impact. Cost-effectiveness, healthcare systems. What were the thoughts there?
This is absolutely critical. Everyone agreed we desperately need long-term, real-world data beyond the controlled setting of trials to figure out the true cost-effectiveness.
Because these drugs aren't cheap.
Not at all. But the flip side is the data from trials showing benefits in things like activities of daily living, ADLs, is encouraging. It suggests these therapies might help people stay independent longer.
Which has huge value, personally and economically.
Immense value. But we need to see how that plays out over years in terms of overall healthcare costs, caregiver burden, and so on. Donna Wilcock raised the tough ethical issue, too. If resources are limited, who gets access? How do we avoid creating bigger disparities?
A really difficult question.
It is. The hope is that by slowing disease progression, maybe delaying the move to more severe dementia, These therapies could improve quality of life and potentially reduce the huge costs of long-term care down the line.
So it's about looking beyond just cognition.
Yes. Measuring functional ability, resource use, psychiatric symptoms, all that needs to be factored into cost effectiveness. There's even some early trial data suggesting benefits for caregivers, which is huge.
That's really important.
The core idea really is disease modification, pushing all those difficult milestones of Alzheimer's further into the future. Janice Smith mentioned how the UK's NICE system evaluates this, really focusing on what outcomes matter most to patients and families.
So balancing the upfront cost against potential long-term savings and quality of life improvements.
Exactly. It's a complex calculation. And as Eric Siemers put it, we should definitely acknowledge how far we've come. These therapies aren't perfect cures, not yet, but they represent truly remarkable progress.
A pivotal moment, for sure.
A moment that could really change how we approach Alzheimer's disease altogether.
This has been incredibly insightful, really digging into where things stand. So just to recap the main points for you listening, Alzheimer's immunotherapy is undeniably a huge step forward.
But plenty of challenges remain. Optimizing safety, especially managing ARIA, boosting efficacy further, figuring out access and delivery, and really understanding the long-term impact. That's the work ahead.
And the field is actively looking at solutions, like combination therapies and revisiting the potential of vaccines, especially for prevention.
Yeah, and some of those aha moments were really interesting. The details of targeting different amyloid types, that brain shuttle idea for potentially safer delivery, and just how critical understanding things like CAA is becoming.
It paints a picture of real, dynamic progress, and the prospect of moving towards prevention offers, well, genuine hope.
Absolutely.
And if all this science feels a bit complex or overwhelming, the key takeaway for you is that this rapid progress, even with its challenges, is charting a much clearer path forward than we've ever had before in Alzheimer's research.
It really is. Which maybe leads to a final thought for you to ponder.
Yeah.
As we get better, as we potentially move towards intervening even before symptoms appear, how do we think about risk?
Risk versus benefit in people who are currently healthy.
Exactly. How will we define that? How will we communicate it? Especially when we talk about potential preventative treatments for asymptomatic individuals. It raises some profound ethical questions for society to grapple with as this science continues to advance.
That's a really important point to consider as we move forward. We definitely encourage you to keep following these developments. Resources like the Alzheimer's Association and major research institutions are great places to stay informed. Thanks for joining us for this deep dive.